Kuelika Mukherjee, a botany postgraduate from Banaras Hindu University, and today I will discuss the light reactions that takes place during the photosynthesis in high. So let's start with what is photosynthesis. If we break this word photosynthesis, photo plus synthesis. Photo means light, photo will mean light, and synthesis means to produce. So, photosynthesis is a physico-chemical process in which glucose or sugar molecule is formed in the presence of light. The entire photosynthetic procedure is divided into two events, that is light reaction and dark reaction. Before we dive into the entire process of light reaction which we are going to discuss today, let us first recapitulate the basic structure of chloroplast and then we will move forward. If we see the basic structure of the chloroplast, it is a two membranes organelle. Okay, there is an outer membrane and there is an inner membrane. There is an outer membrane and there is an inner membrane. Inner to this inner membrane, we have the stroma. Stroma is the site of the dark reaction. So, it is the site of dark reaction. Inside the stroma, we have vesicle-like structure called as thylakoids. So, we have thylakoids, and these thylakoids, these are stacked together. Okay, these thylakoids are stacked together, and this stackings is called as granum. This is called as granum. Singular, it is called as granum. If more than one granum molecules are present, then these are together. So plural form is basically grana, singular form is granum. Okay, these granum molecules are being interconnected via the stroma lamellae. This is the stroma lamellae. So as you know that chloroplast is a semi-autonomous replicating system. It has its own DNA molecule. It is a double-stranded DNA molecule and it has also got 70 S ribosomes. These are 70 S ribosomes. So this is the basic structure of chloroplast. Now, if you see a single thylakoid, okay, this is not a two-membrane structure. It is a lipid bilayer, single membrane lipid bilayer structure, and inside we have lumen. This is a thylakoid. So before starting the light reaction, there is a small concept I want to give you about the photosystems. So what are photosystems? Photosystems are basically, these are the photosynthetic units, okay? Photosystem. Why we call it as a system? Because it is made up of many components. System is something that comprises of many components. So it is basically a photosynthetic unit. What it is? It is a photosynthetic unit. What it comprises of? You will see. It comprises of the reaction center, okay, the reaction center, as well as the light harvesting complex. The light harvesting complex. Or in short, we call it as the LHC. The light harvesting complex is basically hundreds of pigments that are bound to the proteins. So what happens, the reaction center, which is basically the chlorophyll A molecule. What it is a chlorophyll A molecule. Both of these reaction center and light harvesting complex, both of these, they will receive the light energy. And what light harvesting complex does, it receives the light energy and it transfers to the reaction center to increase the efficiency of the reaction center. Now, the photosystem is basically of two types, photosystem 1 and photosystem 2. The basic difference between the photosystem 1, I am writing it in short, okay, photosystem is PS. So, PS1 and PS2, photosystem 2. If you see the difference between the two, the absorption maxima of photosystem 1 is 700 nanometer. 700 nanometer is the absorption maxima of photosystem 1 and the absorption maxima of photosystem 2 is 680 nanometer. Photosystem 1, it is involved in both cyclic and non-cyclic photophosphorylation. Cyclic and non-cyclic. Okay, and photosystem 2, it takes place only in the, it is involved in only in the non-cyclic photophosphorylation. I am writing it in short, NPC, non-photo, NPP, non-photo, non-cyclic, NCP, non-cyclic photophosphorylation. So now that we have understood the basic concepts of the photosystems, we will now see the non-cyclic photophosphorylation. Now break this word photosphorylation. Again, photo means light and phosphorylation means generation of ATP. So in the presence of light, there is generation of ATP that will be further used while the dark reaction. So let's start the non-cyclic photophosphorylation. What happens when the light falls on the photosystem 2? The light falls on the photosystem 2. The electrons of the photosystem 2, they will get excited. The electrons will get excited. So 2 electrons from the photosystem 2 will be released to the next electron carrier, that is the pheophytene. Pheophytene is an electron carrier. It is also a modified chlorophyll A molecule which lacks the central magnesium. So after pheophytene, the two electrons will go to the plastoquinone molecules. These 2 electrons will go to the plastoquinone molecules. Plastoquinone is also an electron carrier and it is a quinone molecule. From the plastoquinone, it will move, the 2 electrons will move to cytochrome B6F complex. The 2 electrons will move to the cytochrome B6F complex, which is also an electron carrier. Cytochrome B6F complex, which is an electron carrier and it is also a proton transporter. So it transports protons from the stroma side towards the lumen side while carrying the electrons. So while doing so, it creates a H plus gradient in the lumen, and while doing so, there will be a high concentration of H plus in the lumen side. Therefore, there will be a proton motive force that will generate for the formation of ATP. This you will better understand in the chemiosmotic hypothesis. So the 2 electrons will be further carried up. The 2 electrons will be further carried to the plastocyanine molecule. The plastocyanine molecules are also electron carriers, but these are copper containing molecules. Copper containing mobile electron carriers that will move from here to the photosystem 1, and it will transfer the 2 electrons to the photosystem 1. These 2 electrons will reach the photosystem 1. The photosystem 1 will also get excited and it will also release the 2 electrons to the next electron carrier of the photosystem 1, that is an iron sulfur cluster. So FES, it will transfer the 2 electrons to the FES molecules. From the FES molecules, the 2 electrons will be transferred to ferredoxine molecules. The ferredoxine molecules will accept the electrons and it will transfer the 2 electrons to FNR. This is very important step. FNR is ferredoxine NADP reductase enzyme. What this enzyme will do, it will reduce the NADP molecule, NADP plus molecules into NADPH. So a reducing power is generated in the non-cyclic photophosphorylation, and here an ATP molecule was also generated. So ADP plus PI will generate an ATP molecule. So in non-photophosphorylation, a ATP molecule and the NADP molecule both are formed. This non-cyclic photophosphorylation takes place in the thylakoid. But will the photosystem 2 continuously lose electrons? It cannot do so. So how will it manage this loss? This loss is managed by photolysis of water that takes place in the lumen. So what happens? The water in the lumen itself, the water molecules, the 2 water molecules will be broken down. That is called photolysis, the lysis of water molecules in the presence of light. 2 water molecules will be broken down into 4H plus 4 electrons plus, and oxygen molecules will be liberated. The 4H plus are used by the ferredoxine NADP reductase. The 4 electrons, it moves to the photosystem 2 to manage its loss. So now the 4 electrons will be accepted by photosystem 2 to again carry this process. Now you can see the shape of this non-cyclic photosphorylation. It looks like a tilted Z, right? That is why it is also called as the Z scheme. This was the entire non-cyclic photophosphorylation. Now we'll see the cyclic photophosphorylation. Now let's see the cyclic photophosphorylation. As the name suggests, it is a cyclic process. In the cyclic photophosphorylation, first of all, the site of cyclic photophosphorylation is the stroma laminae. Okay, where it takes place in the stroma laminae. Cyclic photophosphorylation, only ATP is produced. There is no formation of NADPH. Okay, so now let's see the formation of this cyclic photophosphorylation. Only photosystem 1 is involved in cyclic photophosphorylation. Only photosystem 1 is involved. No photosystem 2 is involved. So from the photosystem 1, when it receives the light, the photosystem 1 will get excited. It will lose its 2 electrons and it will give to its first primary electron acceptor, that is the iron sulfur protein. From the FES, these 2 electrons will move to the ferredoxine. This will move to the ferredoxine molecule. And remember, there is no FNR here, there is no ferredoxine NADP reductase. So this FT, that is ferredoxine molecule, will pass on this electron to plastoquinone. This electron will be passed on to plastoquinone. Plastoquinone molecule. From the plastoquinone, these 2 electrons will be transferred to the cytochrome B6F complex. Cytochrome B6F complex. From the cytochrome B6F complex, the 2 electrons will be transferred to the mobile carrier, that is the copper containing mobile electron carrier, that is plastocyanine. plastocyanin, these two electrons will go back to the photosystem 1. So, in this cyclic photophosphorylation, there is no loss of electrons. The electrons that were released are coming back to the photosystem 1. So, tell me, is there a need of photolysis of water here? Because there is no loss of electrons. No, right? There is no need of photolysis of water. So, we can see that in this cyclic photophosphorylation, only ATP is produced. No NADPH is produced. And there is no photolysis of water, so there is no oxygen evolution. We will now quickly see the difference between the non-cyclic and the cyclic photophosphorylation. Now let us quickly recapitulate what we studied in the light reactions. So we started from the basic structure of chloroplast, where we saw that stroma is the site of the dark reaction and the grana and the stroma lamellae are the site of light reactions. After that we studied about the photosystem, about the photosystem 1 and photosystem 2. That photosystem 1, the absorption maxima is 700 nanometer, and in the photosystem 2, the absorption maximum was 680 nanometer. The photosystem 1 was involved in both the non-cyclic and the cyclic photophosphorylation, whereas the photosystem 2, it was only involved in the non-cyclic photophosphorylation. Now, we also saw the structure, the phenomena, how the non-cyclic and the cyclic photophosphorylation takes place. Now, let us recapitulate once what was the basic difference between the non-cyclic and the cyclic photophosphorylation. So, the first difference is the site where it takes place. Non-cyclic photophosphorylation, it takes place in the thylakoid membrane. It takes place in the thylakoid membrane. And the cyclic photophosphorylation, it takes place in the stroma lamellae. The non-cyclic photophosphorylation, both photosystems are involved. So, both photosystem 1, PS1 and PS2, photosystem 2, these are involved. In case of cyclic photophosphorylation, only photosystem 1 is involved. Only PS1 is involved. In non-photophosphorylation, the products were ATP and NADPH. In case of cyclic photophosphorylation, only ATP is produced. In non-cyclic photophosphorylation, we saw the photolysis of water. In cyclic photophosphorylation, we saw no such process. So there is no photolysis of water. During the photolysis of water, we saw that oxygen was evolving. So there is an oxygen evolution. And in cyclic photophosphorylation, there is no oxygen evolution. So this was pretty much it about the light reactions. This brings me to the end of the lecture. I hope you liked it.